solenoid structure comes first, then the rosette structure. So the rosette is actually made up of the DNA in its solenoid structure. So that cylindrical structure is then going to be looped around. We already talked about that. Okay. The loops here are going to be different sizes. And they're going to be different sizes because what may have to be happening at that time, especially in transcription. So if the loop is larger, it's going to be more accessible to all that transcription machinery. Those would be regions that would be needed to be transcribed. If the loops are smaller and closer to those scaffolding proteins, those are regions of the DNA that don't need to be accessible.
Now, would that be because there are more nucleosomes in the smaller loops?
It could be. It could be due to the number of nucleosomes, but it may not be. And I don't know the right answer.
Okay, so just more transcription in a bigger loop.
So they're not going to be uniform size. Okay. So, now, replication. You guys remember DNA replication? 2 parental strands get replicated, make 2 duplexes, 1 parental, 1 daughter. Okay. Well, as DNA replication happens, the nucleosomes have to be put back into those new duplexes. Okay? What would happen if that new DNA was not packaged? What if it was just made? How much DNA did I tell you? If it was naked, it would be stretched out. 6 feet. Would it fit in your nucleus?
No.
That's why it needs to be packaged. So as soon as the DNA is being made by DNA replication, it needs to be packaged so it fits still in that nucleus. Because now you have twice as much genetic material. It needs to be packaged efficiently. Okay? So here's the replication machinery. Okay, helicase, DNA polymerase. Replication fork moving this way. Here's the 2 parental strands. They are already wound by nucleosomes. Yeah? Okay, so down here, these are each 1 parental, 1 new strand. Okay? As the new strands are made, okay, they have to be packaged in nucleosomes right away. The histones used in the nucleosomes during replication are part of the old histones with new parts also. So those old histones have to be removed from the DNA to make it accessible for the replication machinery. Yeah? You gotta remove this in order to get to all the DNA to replicate it. Those histone parts will then be used down here to help package. So they'll be recycled. Yeah? Okay. But there's not enough histone parts from up there. So these new nucleosomes, these new octamer of histones, are going to be part old and part new parts. Some of them will be old histones, some of them will be new histones. But it's a recycling mechanism so that they don't have to destroy all of the old parts and make all brand new. Does that make sense? Just recycling. Okay? Recycling's always good, right? Okay. Because they're recycling some of those old parts, sometimes, oftentimes, those histones have chemical groups added to them. If there are chemical groups on those, there's going to be chemical groups over here if they were recycled. So the chemical modifications to the histones are going to be maintained even in the new daughter strands. Okay? You guys look really lost. Are you really lost, or is it just Thursday afternoon?
Thursday afternoon.
Okay. Just checking. Okay, so the chemical modifications. These are also grouped as epigenetic modifications. Epigenetic modifications are additions of chemical groups. They in no way impact the DNA sequence. There is no change in bases, there's no insertion of bases, there's no deletion of bases, there's no change in sequence. Okay? These are chemical groups that are added to, in this case, the histones, to change what's going on the chromosomal level. Okay? So if methyl groups are added to the N-terminus of the histones, okay, methyl groups tighten the DNA packaging. Okay? They're going to reduce the availability of that DNA for transcription. So methyls on the histones are going to tighten up the overall structure. Okay. Acetyl groups on the histones are going to do the opposite. They're going to loosen the affinity of the DNA to the nucleosomes. They're going to make the DNA more accessible for transcription. Okay. And I kind of think of this as a nicely choreographed dance, that constantly there's going to be methyl groups added, acetyl groups added, depending on what the cell needs at that time. Okay, do we need that gene accessible, or do we need the gene inaccessible? Okay, because, as I said before, those old histones are recycled. If these old histones have methyl groups on them, these will now have methyl groups on them. So when they're recycled, the methyls and the acetyl groups on the N-terminus are not removed. So if this needs to be tightly compacted, it will still be tightly compacted. If it needs to be loosely compacted, it'll be loosely compacted. Okay? Because of the chemical modifications from up there. Okay? So putting these modifications on the histones and exposing the promoters and the rest of the the gene sequences to transcription machinery is called chromatin modeling. You are changing the proteins bound there because you need some function. Okay, ways to differentiate chromosomes. Cytogeneticists, that's the name given to scientists, geneticists, who look at chromosomes. I am not a cytogeneticist. But cytogeneticists are very good at looking under a microscope and actually being able to organize all the chromosomes based on their size, their shape, and their banding patterns, which we haven't talked about banding patterns yet. Okay, so eukaryotic chromosomes, they are linear. These are examples at metaphase, which is when cytogeneticists look at the cells because they're the most condensed at that point. Okay? Or at the chromosomes because they're the most condensed. So there are 4 different groups that they put chromosomes in. They're all based off of where the centromere is located in relation to the arms of the chromosome. So metacentric chromosomes, the centromere is in the center. Okay? Each arm is about the same size. So metacentric, in the center. The 2 arms on either side of the centromere are given different names. The P arm and the Q arm. Okay, it was given the name P arm for petite. So that's your easy way of remembering P, petite, small. Okay? Q is long arm. In submetacentric, the P arm is much shorter than the Q arm. Okay? In acrocentric, the P arm is itty bitty bitty bitties, but it's still there. Okay? So submetacentric is kind of between the acrocentric and the metacentric. Okay, it's getting, the P arm is getting smaller and smaller. Telocentric is when the telomere actually coincides with the centromere. Remember, telomere is on the end, so telomere is usually here and here. Telocentric, telomere and centromere are right next to each other. We do not have any telocentric chromosomes in our genome. Ours are mostly acrocentric, submetacentric, and metacentric. I think Drosophila or mouse have telocentric ones.
It's always a
I have a
stupid
question
flies.
on the satellite. Is that part of the P arm, or is that,
It is.
okay.
It looks like it's kind of hanging off.